I'm gonna start the chapter number 12 from the book JUNQUEIRA. Is about blood. Blood is a specialized connective tissue consisting of cells and fluid extracellular material called plasma, propelled mainly by rhythmic contractions of the heart. About 5 liters of the blood in the average adult moved unidirectionally within the closed circulatory system. This is the so-called formed elements circulating in the plasma are erythrocytes, red blood cells, and leukocytes, white blood cells, and platelets. So in this paragraph, it's about telling about the blood. What is a blood? It's a plasma. And what in the fluid plasma, what is inside the fluid is cells, is erythrocytes, which is red blood cell, leukocytes, white blood cells, and platelets. When blood leaves the circulatory system, either in the test tube or in the extracellular matrix surrounding blood vessels, plasma protein reacts with one another to produce a clot, which includes formed element and a pale yellow liquid called serum. So serum contains growth factors and other protein released from the platelets during clot formation, which confer biological properties very different from those of the plasma. Collected blood in which clotting is prevented by the addition of anticoagulants. Examples are hairpin or citrate can be separated by the centrifugations into the layers that reflects its heterogeneity. Sorry. Erythrocytes comprises the sedimented material, and their volume normally about 44% of the total blood volume in the healthy adults is called the hematocyst. The straw-colored, translucent, slightly viscous supernatant comprises 55% at the top half of the centrifugation tube in the plasma. A thin gray-white layer called buffy coat between the plasma and the hematocytes, about 1% of the volume, consisting of leukocytes and platelets, both less dense than erythrocyte. Blood is a distributing vehicles transporting O2, carbon dioxide, metabolites, hormones, and other substances to cells throughout the body. Most O2 is bounded to hemoglobin in erythrocytes and is much more abundant in arterial than venous blood. While carbon dioxide is carried in a solution as CO2 or HCO3 in addition to being hemoglobin bounded. Nutrients are distributed from their sites of synthesis or absorption in the gut, while metabolite residues are collected from the cells throughout the body and removed from the blood by excretory organs. Hormone distribution in the blood permits the exchange of chemical messages between the distant organ regulating normal organ function. Blood also participates in heat distribution, the regulation of body temperature, and maintenance of acid, base, and osmotic balance. Next is leukocytes. White blood cells have diverse function and are one of the body's chief defense against infection. These cells are generally spherical and inactive while suspended in circulating blood. But when called to sites of infection or inflammation, they crosses the walls of venules, become motile and migrated into tissues and display their defensive capabilities. Display their defensive capabilities. Next is composition of plasma. Plasma is a aqueous solution with a pH 7.4 containing substance of low or high molecular weight that make up 7% of its volume. The dissolved components are mostly plasma proteins, but they also include nutrients, respiratory gas, nitrogenous waste, hormones, and inorganic ion, collectively called electrolytes. Through the capillary wall, the lower molecular weight component of the plasma are in the equilibrium with the interstitial fluid of tissues. The composition of the plasma is usually an indicator of the mean composition of the extracellular fluid in tissues. There are major plasma protein including following. So number one is albumin, the most abundant protein. Plasma protein is made in liver and serve primarily to maintain the osmotic pressure of the blood. Second protein is globulin, is alpha and beta globulins, is also made by liver and other cells, including transferrin and other transport factors. Fibronectin, thrombins, and other coagulations. Next is factors, lipoprotein and other protein entering blood from tissue. Immunoglobulins, antibodies, or gamma globins, sorry, no gamma, secreted by plasma cells in many locations. Fibrogens is the largest plasma protein, 314 kD, also made in the liver, which during clotting polymerizes as insoluble cross-linked fibers of fibrins that blocks blood loss from small vessels. Complement protein, which comprises a defensive system important in inflammation and destruction of microorganisms. So in this paragraph, composition of plasmas shows about how much volume it has, what is the pH of plasma, what nutrients include in the electrocytes, electrolytes, what include in the electrolytes, where it is formed, what is its density, what types of protein in it. And there is one, this is a table, it explains this paragraph that I read above. So composition of blood, as you can see, the orange cream color component that we get after centrifugation. The top layer is a plasma, which is 55% of whole blood, which contains water. It has 92% of water, 7% protein, and these are the types of proteins with the percentage and other solute by weight. And there is a small buffer coat between these two erythrocyte and plasma layer. Is a buffer coat which is 1% of the less than 1% maybe of the whole body. Platelets, it includes leukocytes, neutrophils, lymphocytes, monocytes, xenophils, basophils, and there is a last layer is erythrocytes, which is 44% of whole blood, and these cells in it. How do we get this 3 layers? A tube of blood after centrifugation center has nearly half of the volume of represented by erythrocyte in the bottom half of the tube, and the volume is called hematocrit. Between the sedimentation erythrocytes and the suprarenal light colored plasma is thin layer of leukocytes and platelets are called buffer coat. It's called they are telling what the each section is called. So the red one, bottom one is called hematocytes, hematocyst. The bottom layer is buffer coat. The concentration range of erythrocyte, platelets, and leukocytes is normal blood are including here, along with a differential count or percentage range from each type of leukocyte presenting in the buffer code. A cubic millimeter of blood is equivalent to the microliter. Complete blood count CBC values in these chapters are those used by the US National Board of Medical Examination. Basically, this book is JUNQUEIRA'S, so that's why he's in this book is explained like this. This graph shows the oxygen pressure in the blood vessels, and we can see that in venous, when the blood is in the vein is 40% oxygen. When the blood went to the lung capillaries, its concentration increases because from lungs we breathe in, so air comes in the blood, so O2 comes in the blood, in the capillaries, and then arteries is a little bit less than that, and then slowly when it divides into capillaries and veins then slow down because the oxygen is distributed in the whole body. So the amount of O2 in the blood or the O2 pressure is highest in the arteries and lung capillaries and decreases in the tissue capillaries where exchange of O2 and CO2 occur between blood and tissue. Thank you very much.